So this video is just going to be talking about CRISPR-Cas9 in oncology. So first, we should just review what oncology is, and it's just the study of cancer. Cancer is a horrible disease. It's where cells divide uncontrollably and they spread, and this is caused by changes to the DNA. It is a leading cause of death globally, where 1 in 6 deaths in 2020 were from cancer. So, related to this is precision medicine, more specifically, precision oncology. And this is just the matching of treatment to the patient based on their molecular biology. So, we're going to talk more about this, but basically, the researchers and doctors can look at a patient's DNA, give them treatment, look at their biomarkers, and just see what what are the specifics of their cancer and what is the best way to treat it based on their DNA? And again, we're going to talk more about this. So now that that's out of the way, we can talk about CRISPR-Cas9. So CRISPR is a family of DNA sequences that are naturally found in prokaryotes. And CRISPR-Cas9 is an enzyme that can cut DNA. So a guide RNA finds the appropriate segment of DNA and it brings CRISPR-Cas9 to it. So scientists figured out that they could make their own guide RNA to make CRISPR-Cas9 edit virtually any gene they want. The scientists can knock out, activate, or interfere with genes and see how the cancer and drug resistance is affected. Then, based on the patient's reaction, they can figure out exactly what treatment is best for the patient. And T cells can be edited as well to better attack the cancer. So, with cancer, immune cells or T cells can't identify that the cancerous cells are bad, and so they don't attack the cancer. But scientists can extract the T cells from the patient, edit them outside the body, and then infuse them back into the patient so that the T cells can then attack the cancer. So this is still experimental and it won't work on all types of cancer, but it's definitely promising. So, like I was mentioning before, here are the T cells attacking the cancer cell after the treatment is done. So then, of course, with a lot of things, there are pros and cons. This is new, relatively speaking. It's still experimental, still being developed, so there's definitely pros and cons. So obviously the pro is what I've been talking about. It's a more effective way to treat cancer. Um, and like I said, it's because it can specifically be designed for a specific patient based on their exact DNA and genetic makeup. Um, but there are cons. So, one, it just may be hard to get all of the cancer. And this is just because cancer is so aggressive. So, even if you even miss just a few cells, they can just, multiply again and then you'll get a relapse. So, they are working on just making the treatment more effective and getting all of the cancer. And then there could be some unwanted effects from the treatment. People are worried that the CRISPR-Cas9 technique could affect the wrong cells, or not cells, the wrong genes. So, like it could turn on a bad gene, turn off a good gene. So they're working on that. And then there are morality issues, just more broadly with gene editing. People have issues with embryo editing, possible like bioweapon usages with edited DNA and everything. So there are definitely things that need to be figured out, but overall, this is really promising science, and it could be really beneficial and life-changing for a lot of people. So I'm excited to see where it goes.